Okay, grade 11, welcome to our last unit of the first year of the DP. This is not a content unit as far as learning new biological facts or about a specific field of biology or branch of biology. We're going to be instead looking at experimental design, statistics, and you're going to do a mock IA. So this is more about applying what you've learned and refining those skill sets you need to do a really good IA next year, and just prepare you better for data analysis questions and that kind of stuff as part of this course. So we have 12 lessons long, and you'll do your mock IA kind of as you go. I don't plan on having a unit test for this because instead we'll have our final exam. We'll include some of this stuff, and you'll have several assignments throughout the unit. Okay, so today's lesson is Introduction to Experimental Design. And what I'm going to ask you to do for your format for note-taking in this unit is you need to make a table, or if you're using on paper, to split your page in half. And the one half, you're going to make notes on experimental design. So whatever the lesson is, you'll take your notes here. And then your mock IA, you're going to find that things I say or things that you read about or things you do kind of give you ideas, or you have thoughts about, oh, I should make sure I do this for my IA. Those moments are really precious, so you need to write that information down as it comes to you. So I'm forcing you guys to do the split page note-taking style. Some people do kind of like Cornell notes. You have what I have on the screen and what I'm saying here, and your own thoughts, how it relates to your IA on the side here. And when I'm checking your homework completion, I want to see this format. If you find that you don't need exactly half and half, you can do this part a little bigger, like 3/4 and 1/4. It's up to you. If you're doing it digitally, I recommend you make a table and put half and half in each one, and that way you can keep track of it that way. But you really want to have a space dedicated to your mock IA and your thought process as you develop your ideas. That way, as you think of those things, you can write them down immediately, and you can go back and find them later on. Okay, so let's start by talking about research and how research has developed over time and kind of how research has changed to prepare you for what we're doing in this unit. So how we define research today, and this is a very nice definition I found, is that it's an undertaking intended to extend knowledge through a disciplined inquiry or a systematic investigation. So we're looking to improve and extend our knowledge about some topic, and of course in this class it's going to be about biology, and we're using sort of disciplined inquiry or some sort of systematic investigation. So it's organized, it has steps, it has a process to it. But this idea of trying to answer questions, of trying to figure out, build knowledge, and discover things is not a new thing, right? Humans have been doing this for tens of thousands of years, as long as humanity has been around. Trying to figure out how does this work, or where can I find this, or why does that do that? And it's been changing. How we do it has changed over time, but I think that happens as not. So in later societies, kind of beyond hunter-gatherer, beyond early agricultural societies, we have, for example, in Rome. So this is the School of Athens in Greece here. The School of Athens painting, really famous. That's in Rome. If you have a chance to see it, I think it's in the Sistine Chapel, and it's just really amazing. I actually got to see it in real life, and I can't remember where it was in Italy, but it was really cool. So the idea that we have all these philosophers, these thinking about questions about nature, and they did what are called thought experiments. So thought experiments is an idea where you have some sort of theory, some sort of hypothesis, if you will, an explanation for something in nature, in the world, and then you think about, if that law was true, what would you expect to see in the world? So for example, let's say you have the idea that tiny things, microbes, can make you sick, which they never would have thought of, there's one they actually did think of, that reproduction, babies are made because a man plants his sperm in a woman's menstrual blood. So if that was your thought, you would look for evidence for that. Well, you know that when males ejaculate their sperm there, you know that females produce blood, so that seems logical to you. There's no actual experiments done, no real data collected. It's more just like, do you have observations from your life that support your idea? So that was how it started way back when. And then in later time, in the 1700s, so 1600s or the 17th century, it was adapted and formalized into what we call the scientific method today. And Sir Francis Bacon did this, but he wasn't the only one to do this. He's kind of the first one to really publish and write out a formalized how to do science kind of guide. But Galileo was really big in research, and Copernicus as well. Both were big influences on Francis Bacon, and then Newton also further developed after Bacon more ideas on how to collect and do good research. Okay, so here's a picture of Newton doing research here. And he was a big factor in how we changed, doing many trials of the same experiment and looking for really reliable answers to questions, which is a lot easier to do in physics and in chemistry than it is in biology. Biology is very much harder to control than the other branches of science. You'll learn that doing your IAs. I hope you've learned a little bit throughout this year, especially with our ventilation rate lab with humans. So they got better and better at making the data more reliable, understanding better what their experiment was trying to find and were they actually controlling the various variables, all the kind of stuff. But the ethics part of research took a really long time to get to a decent place, and it still needs work today. Do you remember Florian Chain's experiment? We talked about this earlier about their experiment on antibiotic, about penicillin. If you can remember back, it's talking about the fact that they used, 6 mice total or 12 mice total, and they got them all infected with a bacterial infection. They gave half of them penicillin, half of them not. The ones who had penicillin lived, the ones who didn't have penicillin died, and they proved that penicillin was an antibiotic. And today, that would never suffice. It's not enough data. But like I said, it's a lot harder to collect sufficient data and numbers in biological experiments than it is in other physical science experiments, especially with living things. The ethical part, here's a good example of this. I think HL, we talked about this with immunizations about topic 11. Edward Jenner. So he's known for making, in the Western world, for making one of the first vaccines, and he developed the smallpox vaccine. And he did this in such a way that is very questionable ethically. And what he did is he realized that milkmaids who were exposed to cows who had cowpox did not catch smallpox. So he thought that was kind of an interesting observation. And then what he did to test this idea is he took pus from the blisters on cows who had cowpox, and then he injected it, or scraped, not really injected, but scraped a child's arm and put the pus into their arm, and then exposed that child to smallpox to see if they got smallpox or not. And of course, they didn't get smallpox. They got a little bit sick, but they didn't get fully sick because their body had the antibodies to fight off smallpox. The very first vaccine ever in the Western world. There's evidence that they have done it as well in ancient China and other ideas for doing similar kind of ideas much before Edward Jenner did it. But this is super questionable ethically. We'll talk more about this and other examples of ethical issues in research. Okay, so here's a series of cartoons I found, which are really great for kind of summarizing how research and ethics in research has progressed over time and giving some good examples of this. And I want you guys to really try and understand the history of ethics, because you need to understand where the concerns for consent and care for your specimens that the IBO finds so important, where those come from. Okay, so the medical technologies we have today and all the abilities we have to heal people and help people, that took a lot of work and a lot of sacrifice, not just from doctors and scientists, but from patients, human test subjects who are willing to sacrifice themselves or be a guinea pig, if you will, to potentially help develop a drug or a treatment that could help other people in the future. And that's something people often forget about, is that it's not just doctors and scientists who did these things, it's also people they worked with who helped develop these technologies we have today. There's a really long history of this, and for most of the history of using human participants or subjects in medical research and biological research, the participants didn't have any choice in this. It wasn't really consent. So for example, here we have in ancient Rome or ancient Greece, we have a prisoner who's going to get executed off with his head, and then a scientist is like, hey, can I borrow him first? because he wants to do some experiments on this guy who's going to die anyway, is the thought. He's going to be executed. Might as well do some experiments on him first, trying to learn more about the human body. So at this time period, this was very common to do what's called a vivisection, where they would take someone who was still alive and start to dissect them. So vivisection means still living dissection. And there was still the Hippocratic Oath, this idea that physicians have a moral code. If they're practicing medicine, they have to do no harm. Their idea is they're only going to help people to the best of their ability. This oath existed at that time. But when people were pursuing medical breakthroughs, scientific breakthroughs, looking to learn things, they often kind of forgot about this idea, especially if the people that they were working on or using as subjects they thought were lesser people for some reason. For example, this famous guy, Herophilus, was the father of anatomy. He did lots of vivisections, but he did them on convicts. And because they were convicts, therefore it was more thought to be okay, which is, of course, absurd. But they would do vivisections to try and learn about how the human body worked. And if we continue, Jenner's big discovery here, where he looked at the smallpox vaccine, he realized his cow has cowpox. The milkmaid doesn't have smallpox or cowpox. How is that possible? Why isn't she dead if she's exposed in a village full of smallpox? So his first test subject was a child, an 8-year-old subject named James Phipps, and he was the son of Jenner's gardener. And we don't have any official records of this, but the chances that they got informed consent and explained to this child what was going to happen to him and got his permission to do this to him is pretty darn slim. The idea that Jenner would inject him or kind of give us scrapes and then give him the exposure of the cow's pus from an infected cow, that he inoculated him with it tons of times, like over 20 times to help him test him. And he did other children as well, including his own son eventually. And that's totally unethical that this child who has no idea what's happening to him and can't really say no is being through medical research. That being said, without Jenner's vaccine, millions of people would die from smallpox. So you can see where it gets really easy to rationalize doing tests on humans because of the potential benefits. Okay, at that point too, in time in history, people could do a lot of different things that were more or less kind of creative medical testing. For example, people used to use leeches a lot for medicine, thinking they could leach out the bad sickness, just suck the blood out from the person. And people would just try it out on someone like, oh, they're dying anyway, they're sick, we don't know what to do, let's try this and see if it works. And they would kind of take notes and try to learn from it. But there wasn't a lot of rules or guidance on how to do that. So we really kind of ignore these kind of unwilling test subjects over history. We don't think about them as being a big part of it. But at one point in history, how it's a huge amount and such a horrible way that it got the world's attention and it really began to change the way we look at medical research. And that was in World War II. So the Nazis did crazy, brutal human experiments. They did a lot of twin experiments, trying to see if they did something to one twin but not to the other, how would that affect their health, so the effect of genetics versus experiences, just trying to learn, because they thought of their subjects, if they were in a concentration camp, whether they were Jewish, whether they were homosexual, whether they were a Roma Gypsy, whether they were of any particular group that the Nazis decided were lesser humans, they thought of them as being worthy of being able to just experiment on them. If they died, that's fine. We'll pick another one, try again. So this happened at such a crazy level that eventually when World War II ended and the Nazis were prosecuted at the Nuremberg trials, they decided on some rules for medical research on a global scale because of the severity of this and how intense it was and kind of how quickly it all happened together, and it was so public that these trials that it was the stimulus that was needed to have an actual formally written decision about how we were going to regulate medical research. So this is called the Nuremberg Code, And it stated some pretty simple basic ethical things, that the consent had to be voluntary for the human subject. They had to be voluntary consent. That the experiment conducted on humans had to be conducted so avoid any unnecessary physical and mental suffering and injury. So before this, that was not really a thought for people. It was more about what can we learn that's more important, especially if they could find a way to think about the subject as a lesser human for whatever reason. But American researchers ignored this almost right away. The code was more for barbarians, it wasn't for them. They were above that, they didn't act that way, they weren't Nazis. So it's not applicable to them because they were civilized people. But they did a lot of experiments still on really vulnerable populations, including prisoners, mental patients, poor Black people, are very often used in subjects and studies. So an example of this is a study that began in 1932 and lasted for a very long time, to the 70s, was a Tuskegee study. It's a famous study for its ethical horribleness. It had 600 Black men involved with it. 300 of them had syphilis, and 201 did not have syphilis. And they wanted to see what would happen if someone got syphilis and didn't get treated. And they were looking at how would syphilis look in Black people compared to white people, and is there a big difference here? So they took patients who had syphilis, who didn't have syphilis, and they never really told them what they were doing to them. They just said they were treating them for bad blood. They kind of falsified what the purpose was, thinking those people didn't need to understand and didn't deserve to understand what was happening to them. It didn't matter. Even though they put in the study, they didn't give treatment to them. So when we discovered penicillin was a cure for this in 1947, remember the study began in 1932, so 15 years after it began, they had a cure for syphilis as penicillin, and they had a list of all these people who they knew had it. They did not give them the cure. The study eventually ended in 1972, so it lasted, 38 years, something like that, after a New York Times article published exposed the horribleness of this study. And about 100 people had died from untreated syphilis as part of this study, and many of those subjects gave syphilis to their wives, and their wives and had babies who had congenital syphilis. And the idea was it didn't matter. These were lesser people. The science was more important. It was worth the sacrifice of people, even though it was an unknown sacrifice. They didn't do it knowingly. The participants didn't know what they were getting into because they were doing it for science, for people in general. So then after that, in 1964, we had these further publication on ethics. After the Nuremberg Code, the next one was the World Medical Association published a declaration, which is the Helsinki Declaration. And they were talking now more about specific groups that are going to be more vulnerable groups. So maybe there are people who are in vulnerable groups because they are more vulnerable to society, people of color, children, people who are mentally handicapped, people like orphans, and people who have lower income levels. All these kinds of people who are more likely to be targeted by medical researchers. And the Helsinki document talked about the importance of protecting those people even more so than people who have more privilege and are therefore less likely to be targeted. At the same point, this was being written and agreed upon on a global scale. So another messed up experiment happening in the United States at Willowbrook State School, and this is for children with mental retardation. We wouldn't say that word today, but children who are mentally delayed for whatever reason. And these two scientists are working on studying hepatitis. So they gave these children hepatitis to see how they could treat it. And at one point, it's even written down that someone heard one of the doctors, Dr. Krugman, said that he might as well purposely infect 60 children with hepatitis because they would probably contract it anyway. And it's this idea that lesser people, who people think of as lesser, for whatever reason, are welcome subjects to be tested upon. Another example from 1951 to 74, about 1,000 inmates in prisons were subjected to toxic chemical exposure. So we had one scientist in a prison, a doctor, who decided he wanted to learn how different chemicals affect people's skin. So what he did is he just took inmates and put the chemicals directly on them and then looked at the effects and did research that way. These inmates had no consent. They didn't they were being harmed. They did not know what they was getting into, all this kind of stuff. So this was happening until 1974, pretty recently. And then finally, in 1983, the US kind of got its act together, and medical schools and hospitals had these federal, national level regulations set up with ethical committees for any sort of research. And they had to have higher standards for human research starting in 1983 in the United States. So when I did my master's, I did some research on international education, and I had to submit my research to an ethics board. And I was just doing interviews, a focus group, and surveys with students. But I still had to submit it to an ethics board to approve my research. Had the consent forms were correct, everything in such a way that it would be giving informed consent and not putting any stress or risk to my participants at all. If you choose to use humans in your research, you will have to do the same thing. So this kind of fixed it, if you will, in the US and North America, and Western Europe was pretty good for this at this point in the 1980s. But now we have globalization, and a lot of vulnerable populations are existing around the world who are easy targets. So a lot of these less developed countries had less rigorous ethics rules, and so a lot of clinical trials for new drugs, medications, techniques moved to the developing world, what we call the third world, which is kind of a problematic statement in itself. But now, today, more than 60% of clinical trials take place outside the U.S. and Western Europe because it's easier to get, so not necessarily an ethical procedure to go through. So there's less risk of litigation, so being sued. Ethical review is less rigorous. A lot of vulnerable people there aren't in a place to challenge authority. Maybe there's a lot more illiteracy, a lot more poverty. So people are willing to, if you can give them some money to take a new medicine, they're more willing to do that. This is a great example of this is in 2012. In India, there was a lawsuit that was filed because a pharmaceutical industry decided which injuries and deaths from a clinical trial were related to the trial and which ones were not. So the government didn't have investigators to investigate this. The clinical trial, the drug company was allowed to just say, 3% of our deaths were caused by the drug, and we'll offer compensation to those people's families. The other deaths were not. There was some other factor in their health caused them to die. So 3,458 people died in this study, but only 3% were caused by the drug. And when it was found out by this NGO that it was the company, not the government, that decided this, it was a huge leak and a huge big deal because they're not protecting their citizens. This happens in a lot of places in the world, unfortunately. So a big problem with these outside studies is there's not a lot of supervision, and it's not really transparent. So people can choose, scientists will choose, okay, these are the studies where we had favorable outcomes for our drug, and these are the ones where we didn't, so we can publish only these studies and not publish those studies. This is not only misleading for doctors and scientists. They think, okay, well, that's the literature, let's look like it's a good drug, it's working well. It can mislead about the effectiveness, it can mislead about side effects, but it can also mean that future subjects might be subject to more experimentation in an ethical way that's happening, depending on what's happening in the world, might be subject to more risk because the investigators don't know the full extent of what that drug has done. All this information is hidden, so they don't know the results of failed trials, of potential side effects, and they go in more blindly. So there's some NGOs that are really working on trying to fix this problem. So All Trials is one, Bioethics International is one, the FDA is one, the US is working on this, the WHO is working on this. They're trying to get all clinical trials to be registered around the entire planet and making raw data be public. So the biggest one most ahead of this is the EU, and for them, all raw data has to be publicly available, and they have to explain the raw data in a way that's in lay language, so anyone can understand what it means. Let's be able to make informed choices about their own health. So this is a long way of improvement since Edward Jenner is injecting children and vivisections, but we still have a long way to go. There's still people who are vulnerable, who are being targeted, but we have made progress. So when I ask you about ethics, you're doing ethics for your IA, this is the reason why. People have been taken advantage of for thousands of years for medical research and other kinds of biological research, and animals as well. And the IBO does not condone animal experimentation. And any research done with humans, you have to be very careful to make sure that you're not causing any sort of stress, you're not causing any harm, and everything is full of informed consent. So this is why we have to do that. So in your lab, there's just 3 kinds of considerations you have to talk about in your IA, and every lab you should be talking about them. Ethics, is it ethical? Do you have informed consent, and are you causing any undue stress or harm? Environmental, are you producing any pollutants? If you have waste products from a chemical reaction of some sort, how do you dispose of the waste products that are causing pollutant? And safety, are there any dangerous chemicals being used, any sharp implements, hot plates, Bunsen burner, anything else can be dangerous. So we have lab safety, we have the environment, and we have ethics. You have to consider all 3 of those for your investigations, every investigation. Now, if they don't apply, let's say you're doing a study that has nothing to do with humans or animals, you don't have to talk about ethics. It's not relevant to your study. If you're not using anything dangerous, you don't have to talk about safety. And they don't like you to talk about things that are not relevant. So to think about this when you're looking at your experiment choices. Okay, so that's what I want to talk about today. Let's talk about what the IA is and what your task for today is. So the IA is individual investigation. It's worth 20% of your final assessment for both SL and HL students. So the same rubric for SL and HL, same expectation for SL and HL. I can give you advice and guidance to the process, but I cannot edit your work, and I can only read through the entire IA one time, and that has to be the time before you submit your final draft. So your final draft that you're most happy with, you would give to me, and I would read through it. I can give you comments and narrative feedback, but I couldn't make edits. And then you can edit that one more time and then submit it. Throughout the process, you're welcome to ask me questions, so I can help you that way. And I'm helping you by doing this unit, but I can't guide you too much. It's a very tricky line. So once you have your first draft completed, I will read it and give you feedback, but no edits, like I just said. The completed IA should be 6 to 12 pages long, and it needs a purposeful research question and a scientific rationale for it. And that's what we're going to start with, is your research question. So this is the scientific process from the IBO, and we're going to be talking about starting off up here with what do you want to ask a question about. So what do you see on the world around you that makes you curious to ask a question? You can explore the literature, what's already out there about this topic you're interested in. What kind of things might inspire you? And you share ideas back and forth. Your brainstorming is great to do with other people. And once you have this part going, let's in the first few classes this week kind of working on the brainstorming ideas of what your topic might be for your mock IA, and then we'll start to gathering into brain data part of it. Okay, so this is in the document that I've posted along with this lesson on Schoology for you, but the most important thing for your IA is picking the right IA. Okay, you need to take a little bit of time to do this. We're going to work on IA's brainstorming list this week. Have the 10 ideas starting today and kind of narrowing it down as you go. So here's going to be a list of do's and don'ts I'm going to show you guys and stuff from the IB guide. So this is directly quoted from the IB subject guide. So possible IA tasks, a hands-on laboratory investigation, which is not really an option right now unless you have something at home with plants or something like that you can still do virtually. If you can think of something, you're welcome to do so. You can use spreadsheet for analysis and modeling. You can use extracting data from a database and analyzing it graphically. You can produce a hybrid of spreadsheet and database work with traditional hands-on investigation, which is a common one to do, is a mix of both. You can use a simulation as long as the simulation is interactive and open-ended. Some simulations just like predict the ending. Some you can have more open-ended ones. I'll show you an example of that in a second. And some of those tasks will consist of qualitative work and quantitative work, so both descriptive and numerical work. So this is an example of a simulation. You can click the link. that is open-ended. So this is, oh, here it is. This is a simulation where you can change the sea level, the tide level, and the two different barnacles, the orange one, the blue, and then here we have the predator, and you can see that affects the density. So I can change the data here, okay, and then I can let it go for a bunch of time, with all those tides going up and down, and then I can see a data table. So I can use this kind of simulation as a great way. I could do an open-ended investigation where I'm picking what variables I'm going to do. If you want, you can recommend just looking for Googling whatever topic you're in, look for virtual labs and that are simulations. Virtual biology lab has a whole bunch of cool ones you can use. And if you're not sure it's open-ended enough or not for an IA, just feel free to ask me. I'm happy to help you figure that out. So that's one really doable option for our current quarantine situation. So here's a good list of do's and don'ts for picking your IA topic. So let's start with the do's. Make sure you have a specific and focused question. Okay, so for example, how does pollution affect plant growth? Way too general. How does concentration of nitrogen in the soil affect plant growth? Better. How does nitrogen soil affect root density? Much better. So being very specific is the way you want to go. Make sure you have a real personal connection. So you're going to get points for personal connection in the IA rubric. We'll look at that a little bit. So if you don't have a personal connection to the topic, it's not a good one for you. So is it connected to your future career choice, a childhood experience, a hobby, a passion, an interest? Why did you choose that topic? Make sure you're able to get enough data. So at least 5 trials and at least 10 to 30 iterations is ideal. Remember, for trials, we do the exact same thing at least 5 times. Whereas iterations is slightly different things to kind of see how the trend goes. So for example, I have the barnacle one, I have the exact same data 5 times, maybe I would change the tide level from 0.5 meters, one iteration, do that 5 times, 1 meter, do that 5 times, 1.5 meters, do that 5 times, that kind of thing. The 5 trials is so you have enough data to take a standard deviation. If you're doing an experiment in the lab or at home, make sure you have enough materials and equipment to do your investigation. So check your teacher. You need to have sufficient accuracy. Accuracy is an important part of your data collection. Make sure there's other studies that you can compare your findings with. A big part of the discussion is comparing the scientific context. If you don't have enough other studies to look at, it's not going to work. And make sure you have informed written consent for any investigations involving humans. I have a template form you can use to fill. We used it for our ventilation lab if you're choosing to use humans in your IA. Don't do something we already did in class. Okay, don't pick an experiment we did in class and then do it slightly differently or do it the exact same. That's not a good idea. And I will not accept that idea of a topic. So think about the lab practicals we've done, the required practicals for sure, and other ones, and don't pick the same one. Don't experiment on animals. You can observe natural animal behavior, but you cannot do anything that's going to cause pain or stress on any animal, a vertebrate or an invertebrate, or that will affect its health in any way. You're welcome to do things to plants, but not animals. You cannot put investigations that administers drugs or medicines or manipulates the environment or diet beyond any easily tolerated by the animal, by any ranges are unacceptable. You also can't kill animals as part of your experiment. You cannot plan any investigations involving bodily fluids, the risk of bloodborne pathogens. You can use your own saliva or sweat, but no one else's, which makes not a great investigation. You can't plan any investigations that involve bacteria or other microorganisms incubated at or near human body temperature, which is 37 degrees Celsius. Okay, so with that information, hopefully you have enough now to start brainstorming. So your task for today is to look at picking an idea, and you need to come up with 10 ideas for topics. And here's what you should keep in mind. Is there a connection to the course? Is it biology? Be careful, don't go into the psychology range or geography, stick with bio, okay? Can you collect good data, enough data, sufficient data, ethical data? Is there a personal connection and interest in the topic? You're working with a lot, make sure you're interested in what you're researching. So your topic, your task right now is to figure out 10 possible IA topics, write down your notes, and I will give you feedback on those. And over the course of this week, we'll eliminate it down to kind of a top 3 based on what we're learning more about experimental design. Okay, so your homework is to upload the notes. You can see your thoughts. If we're doing the half and half page, split page note taking. and include a list of your 10 ideas for your IA. Okay, and use that checklist document to help you ensure making good choices. It's worth taking the time to pick a really good IA topic. And if you're thinking about IAs right now, think about more than one. You're going to do a mock one now, and your real one has to be totally different when you're going to start doing in August. So keep that in mind as well.